Rose scent exposure, linked to increased gray matter in key memory region. Japanese study suggests daily exposure to a specific rose fragrance was associated with measurable increases in brain gray matter in a small Japanese study, raising questions about how ongoing olfactory stimulation affects brain structure and whether scent-based interventions could support cognitive health. Researchers report that participants who wore a rose essential oil on their clothing for a month showed regional increases in gray matter on magnetic resonance imaging (MRI) compared with a control group that used an odorless solution. Study design and results. Researchers at Kyoto University and the University of Tsukuba recruited healthy adult women and divided them into two groups. 28 volunteers applied a rose-scented oil to their clothing daily for 4 weeks, while 22 control participants applied plain water. All participants underwent MRI scans before and after the exposure period to measure gray matter volume across the brain. Analysis revealed a modest but statistically significant increase in gray matter volume for the rose scent group relative to controls. The changes were not uniform across all brain regions. The most pronounced enlargement occurred in the posterior cingulate cortex, PCC, an area strongly linked to memory, autobiographical associations, and components of the default mode network. By contrast, regions more directly tied to olfaction, such as the amygdala, involved in emotional processing of smells, and the orbitofrontal cortex, involved in odor evaluation, showed little or no volume change over the study period. Why the posterior cingulate cortex? The researchers propose that continuous low-level scent exposure may shift processing demands away from primary olfactory structures and toward networks that consolidate odor-related memories and associations. In people with olfactory decline, for example, the amygdala can become less active, while associative regions like the PCC become more engaged in a compensatory response. A similar dynamic could explain why the PCC showed the largest volume increase here. Persistent scent inhalation may require ongoing memory retrieval and semantic processing linked to the odor. An alternative explanation the team raises is emotional regulation. If the rose scent is perceived ambivalently or triggers subtle negative reactions in some participants, the PCC's involvement in integrating emotional and mnemonic information could drive increased activity and structural adaptation. The PCC's role in memory and emotion makes it a plausible site for such changes. Continuous environmental inputs can redirect neural processing in subtle ways. Implications for cognitive health and aromatherapy. Although greater gray matter volume does not automatically equate to improved cognitive performance, the study touches on clinically relevant ideas. The PCC is known to exhibit early atrophy in Alzheimer's disease, so maintaining structural integrity in this region is of interest for dementia prevention. The researchers suggest that accessible interventions, such as adding a consistent fragrance to clothing, could be an inexpensive, low-risk way to stimulate brain plasticity if future larger trials confirm the effect and demonstrate cognitive benefit. However, the findings are preliminary. Limitations include the small sample size, short duration, single scent type, and homogeneous participant group, women only. Larger, longer-term randomized trials are needed to determine whether scent exposure can produce clinically meaningful effects on memory or reduce dementia risk. Expert insight. This study is intriguing because it links a simple environmental manipulation to structural brain change, says Dr. Maya Thompson, a cognitive neuroscientist, fictional, specializing in sensory memory. But structural plasticity can reflect many processes, learning, compensation, or even subtle inflammation, so replication and behavioral correlation are essential before translating this into therapy. Still, the idea that everyday sensory inputs can shape brain networks offers an accessible avenue for further research. Future directions. Future research should test multiple scent types, include male and more diverse participants, use longer exposure windows, and combine structural MRI with functional imaging and cognitive testing. If subsequent studies confirm both structural and cognitive effects, scent-based strategies could be explored alongside established interventions such as exercise and cognitive training. Conclusion. A short-term study from Japan found that continuous inhalation of a rose fragrance correlated with increases in gray matter volume, most notably in the posterior cingulate cortex. While the results suggest that sustained olfactory stimulation can alter brain structure, they do not yet demonstrate improved cognition or prevention of neurodegenerative disease. Larger, multi-site studies with behavioral outcomes are required to assess whether targeted aromatherapy could become a viable complement to other brain health strategies.